But where's the person? Should I? Yeah. And then, but do I still need to share it in Zoom or is it done?
Well.
All right. Can you hear me? Okay. Yeah. So. Oh. Okay, like this, Daniel? Okay, cool. So, well, my name is Justin van der Hoop. I'm an assistant professor in Wageningen, and I was asked to kind of round the day off. So we started with sampling, and now you have data, you've done your QC, still not fully got rid of it, and now you want to also share it with the world, or at least the parts that you can share with the world. So let me quickly ask all the participants. So who of you already has a methodological data set that you work with or that you've already completed with your analysis? I see the hands are really low. So I'll ask the question once more because it's not that hard, right? Who of you already has a methodological data set that all the data is collected and that you've analyzed a large chunk of it? Yeah? Now I see some hands. Good. So, well, you will pleased to know that to all of you, it still be useful to know what you maybe could have done or maybe still can do. And for all the rest, without hands, if you are starting to plan your experiment, it's actually already good to kind of start at the end of the process. Like, what are the things that other people should know about my experiment in order to understand it and maybe also to reproduce it and or to reuse my data. Okay. So well, that doesn't work. Or maybe I should switch it on. Yes. No, what? There we go. All right. So just before we start, a quick shout out to my group. This is a picture of last year, and I should make another picture soon to accommodate the newcomers of this year in a better way. And yeah, so we are a group working on computational methodological approaches. If I have time tomorrow, or sorry, on Thursday, I will briefly elaborate on what we're doing. But if you're interested, we have a website where you can look. Because now we are at the end of the workflow, and yeah, are you actually fair to your own methodological data and your colleagues, right? So who of you have heard about the FAIR principles? Oh, that is already quite nice. So maybe you can help me what the letters FAIR stand for in terms of yeah, data software good practices. So the F, yeah, the A, sure, the I. Wow. All right, let's see if we got the full strike with the R. Yes. So it seems like yeah, I will repeat everything. Yeah, so don't worry. So it seems like some of you already know this, and that's great, but we will still go over it. So there are several aspects about data and software that are important. So findable is really to make it findable. And maybe before I go into more depth, FAIR is not that you have to share all your data and everything with the world, because especially in the clinical setting, that is sometimes just not possible because of privacy issues and other things like if data of you is getting online that is associated with you, with your identity, and maybe people can then find back medical details about you through your data, that wouldn't be a good thing, at least in my opinion. And I think we are more and more come to realize that could be possible. However, what you could do is separate the data from the identity and only connect it through another identifier that is only known by, for example, the hospital, or in this case, SSI, or other clinical institutes. And then you may only want to share the relevant parameters like age, maybe gender, and country of origin. And you can still, for example, share at least some of the data with the world, because it can be reused and without kind of compromising at the individual level. But findable simply means that people know where the data is. Even if it's behind closed doors, you can still mention where the data is stored. Accessible means, of course, for the data that you can share, that you can actually find it, that you can actually see it and download it. Well, this morning you've seen an example of a repository already, NTMSS2023, that you can access. And then hopefully when internet allows you download the data. Interoperable, that means that, I mean, I can share data, some people shared data 30 years ago, but by now, the file format that they shared it in may no longer exist. And that means you can download the data, but you can no longer use it. Yeah, so interoperable really means that you have to try to do it in a way that allows other researchers to actually also use it in other programs. And reusable kind of connects to that you have to provide the information to make the data reusable. It doesn't make sense to share a lot of data without, let's say, minimal metadata to allow to reuse it. Yeah? I mean, if you dump your plasma data in a repository or on Zenodo without any information, yeah, it may be findable because you can share a link to Zenodo. It may be accessible because people can click on it and download it, but it's certainly not reusable, because if you don't have any explanation, then there's no sense, right? You need this context. Other people, and you've all heard, I guess, about ChatGPT and generative AI models. They already changed the FAIR to findable and AI ready, or fully AI ready. But I guess that if you really would think about it, it also still comes back to the original FAIR acronym. And don't worry, I'm not going to make it boring, but the shares are the slides are shared with you in the Google Drive. So at least you have got an idea about several aspects that are related to FAIR data and FAIR software. But I will focus on the data side today. For example, findable here also already describes the metadata, and we heard the metadata already before today as well. So this is findable, accessible, think about it this way. For example, here the interesting point is that the metadata are accessible, even if the data is no longer available for whatever reason. For example, because the participant in the study said, okay, yes, but after 10 years it needs to go. For example, it could be that could happen, but then the FAIR principle would state that let's say the layer of metadata would still remain there, so that people can see what was there. And then we are at the eye of interoperable. So and I will get back to that. Use terms that people understand, or use terms that are somehow related to each other. But keep that in mind, and we will come back to that a bit later. And then reusable, actually, I already explained during the first slide. So, and my message to you is think about this, and when you publish your work, when you write the manuscript, include a section, a paragraph on data and software availability, depending on the nature of your study, where you kind of try to describe this kind of aspect. Because, for example, when I review papers, then that's the first thing I look at, right? Is there a section where I can find, like, how was the study done? Where's the data? Can the data be shared or not? And yeah, it's so often that I cannot find any information on that. So, and then my reviews are very brief, right? Polite, but brief. That, you know, I'm happy to look at it, you know, but first I need more information, and that's usually what I ask. And then in many cases, a few weeks later, suddenly there is data, and suddenly, but anyway, you know, so that is something to think about. Why not do it directly? Why not? And it's okay if you cannot share, right? If there are good reasons not to share, but then tell the reviewers, tell the world that, right? It's not that difficult. This takes a little bit of time, I know that. So one of the questions I got was to discuss or briefly discuss and highlight the repositories. And you've already seen and heard a few of them today. And I took this picture from a review, a natural product report. I thought it was nice because many of the authors are actually in here today. So, where the advancement in data capturing, also reuse, is discussed. So if you like this topic, then that could be also a good review to read. And this is from the natural product kind of perspective, but with a bit of imagination, you can also apply the same things to the clinical world. And what is actually good to realize, there are several types of repositories as databases. Some accept raw metabolomics data. We know, we heard already of GPS Massive. There's also the metabolites hosted by EBI, and there's also metabolomics workbench, and maybe some others that I now forgot, and then I do apologize to the creators. But I think those three are the main ones that I see, I hear of. And then some people also still upload their data in more generic places like Zenodo and other things. I do recommend to upload it to one of the metabolomics repositories, because also they have the infrastructure to also handle your metadata. This is one reason. And two, other tools often connect to these repositories, so they can then automatically retrieve your data and do fancy stuff with it, which makes your life and your metabolomics colleagues and friends also much easier. So that is raw data. Then we also have spectral libraries, right? UPS has a lot of MS2 data, Mustang. There's a few others you will hear later during the course. That's also, let's say, experimental data. Then we also have the more structured databases, like PubChem and the Atlas. And now it starts to become a bit more kind of complex. And the main thing I want to say is that the context matters here, right? So if you're doing a clinical study, and you know your samples are, let's say, plasma samples, you're interested only in human metabolites and not in the exposome or the full metabolome, then it can help you to kind of restrict the compound libraries you use as potential candidate structures for your study. Makes sense then to use the human metabolite database, because what's in the title, right? It wants to describe or contain all the metabolites in the human. Yeah. But if your aim is to do exposomics using the same data, then I think it's wise to also extend your scope to other databases that could contain pollutants, contaminants, and other types of exposomics, or the food TB, or the food metabolite. So context matters. There's a lot of good reviews out there that describe this context. So read the review, select the databases, and use them as input for tools that need structural structures as way to annotate your data. You will hear that later throughout the week. Yeah, so my title is Anyway, it says reuse of metabolomics use case. And actually, my question to you is: have you ever reused metabolomics data set during your study so far, project? Yeah. So I don't see any participant nodding here, but Scott, what did you reuse a metabolomics data set for? Okay, so but you had your own experimental data, yeah, and then you use another data set to see from another type of like you had what the gut microbiota, yeah, and then you checked in the fecal samples whether you could find any links or direct links. So actually, I also came up with some potential use cases. So and you could try to search for the origin of a molecule that's behind the mass spectrum. Like, let's say you have a complex mixture of the plant root microbiome. Then the question would be where does the molecule come from? Is it produced by the plant or by the microbiota? And in the clinical setting, from our gut, is it from the microbiota, or is it from the food, or is it from the human? I mean, sometimes even that is hard to say when we start to digest things, and the microbiota start to catalyze things, and then we take it up again and do another biocatalysis on it. But you got the main idea, right? So you can try to find out, are there links to other types of sources, so that you can actually start to pinpoint where it is coming from. You can also compare your data to what has already been done, just to see how it lands, right? Is it close to what other people see, or is it completely off? If it's completely off, maybe go back to the drawing table and see, okay, what's going on? Am I doing it right, or are they doing it right? Let's say. You could also imagine that sometimes it's unclear whether your spectrum is actually a real biological molecule, right? I mean, so often we get these unknowns, right? Are they real or are they not? And you can check, then you can run tools like MAST, you may hear about it later from Ming, and see if you see it in other biological data sets. And that would increase your confidence that it's a real thing. And yeah, you can ask new type of repository-wide explorative questions, a bit like Scott already indicated in his example. And this is also a figure from the same review paper that I earlier introduced. And there actually a similar example is provided where all the data in the redo database was considered, and then culture bacteria was selected. So each input file that was annotated with culture bacteria is now selected, and then out there the gut associated bacteria, and then after that we can see, we can investigate the level, the fluctuating level of particular molecules. In this case, they have been associated with biofilm formation, etc., etc. So they have been hypothesized to have a biological role, and this is a way to check if indeed they were present at all, and if we could find differential levels. Well, and so that's as far as we went in this review, but you can see that this type of questions are now starting to become possible to ask, because all the data is together and in a comparable way. And now we come maybe to one of the more complex things on data sharing, and that's the metadata. So ontologies and controlled vocabulary. And what you see here is kind of a way to summarize a lot of information. So basically, if you have a lot of time on your hands, then the best way to share things is using ontologies. Why? Because they have all the properties presented in this slide. And what does it mean? Well, we start at the controlled vocabulary level, and that means that you get a closed list of words or terms that you can choose from. to link to, in this case, your data file. Let's say urine, plasma, or fecal, right? Maybe you work in a clinical institute, and these are the 3 types of data that you have. So that's your choice, right? Urine, plasma, or fecal. So that means if your new collaborator comes with skin cell data, then you have a problem because yeah, it's a closed list. So what to do? So then you need to go back to the drawing table and decide whether or not to add this word to your closed list of words. So the advantage is that it's relatively simple. The database like Redo, reuse of your metabolomics data interface that uses a controlled vocabulary, which is a bit more than only 3 options for many cases. And in that way, because everyone uses the same words, you can start to compare things, right? If otherwise, you still need to compare all the metadata to see if you agree it's the same or not. And then we grow in complexity towards ontology by adding an hierarchical structure. So bacteria, cultured bacteria, or non-cultured bacteria. Then we start to kind of divide this into subterms, spreading out. And finally, if we also add a thesaurus, where we describe a lot of the things better, where we find relationships and do provenance on how things change, then we come towards the ontology. So an ontology is used by, for example, metabolites. And that means that, you know, on the long run, it will probably be a better thing to do, but it takes you a bit more time to put it in. And that is where, well, in the end, you have to choose what to do. However, when you just start the project, you can already think about this and then make sure that the way that you describe your data in your lab book or each other's lab book, that you prepare for a later deposition in one of the repositories. So another thing to do is when you give talks is also to encourage people to share their data. And that is what I also usually do when I give talks. And this is just a slide that I then show with the different types of databases that accept mass spec data and also genomic data, because my group is also doing about integrative omics. So then I always say, come on, share your smiles. And usually people immediately started to share their smiles, so that works. But actually, of course, I also mean the smiles as textual identifiers of structures that you annotate. So, well, thanks a lot for your attention. I hope it was useful. If you want to discuss some of these aspects later in a bit more detail, feel free to come to me or any of the other instructors, because I think most of us will know how to deal with this kind of thing. So thanks a lot. Oh, and I was asked to tell you that the break is reduced to 10 minutes to make up a little bit of the lost time. Yeah, so back in 10 minutes. Yeah, you can ask me a question. But maybe we start the break and then you ask me the question, right?
You're welcome.